All right, welcome back. It's me again, Effie. So this is going to be a short presentation about my project. And again, I'm a PhD student in Copenhagen at the DTU, the Novo Nordisk Foundation for Sustainability. And I'm under the supervision of Tilman Weber, who is known as the anti-smash guy, and Timo, who is in the OpenMS team here at the Kohlbacher lab, and Thomas Ostenfeld-Lassen from DTU Bioengineering. And my project aims to develop new tools for the discovery of new natural products, but metabolomic tools for specialized metabolites, as my title says. Right, so even though we think that specialized metabolites sometimes are magic, they are actually a response, an ecological response, and they're produced by the metabolism of the organism, any organism really, which produces very cool compounds such as spinosine, cocaine, which you've all heard about, and linalool. Linalool is the compound responsible for the smell of lavender, one of the compounds responsible for the smell of lavender anyway, and it's a terpenoid. Yeah, so organisms produce all these compounds as a response for pollination, survival sometimes, competition and predation. And it could be that they produce those specialized metabolites and we use them as antibiotics, pigments, or different odors. Right, so some examples of specialized metabolites that we use every day or on our lives anyway, is different antibiotics like streptomycin, which in the past has been used for treatment of tuberculosis and the plague. And as the name says, it was produced by streptomyces. It was a very, very important antibiotic. Of course, penicillin, you've all heard of penicillin, probably you have taken some in your lives. Discovered by Alexander Fleming, and neomycin, also just a few examples of antibiotics. Then it's there are bacteriocins that are also used as preservatives in food, and Germany is really crazy about this cheese, like raclette cheese, I think. And in the wax of this cheese, there's this compound called natamycin, so it preserves the cheese. Just don't eat it if you could avoid it anyway, because it's antimicrobial. So yeah, cool. Then nutrients, actually vitamin B12, that's only produced by bacteria and archaea. Very, very important for our DNA synthesis. We also use a lot of specialized metabolites in cosmetics like curcumin, which is from turmeric, and it has this yellow bright color. Essential oils that could be could have these characteristics of odor or be used as antiseptics. Then analgesics. Okay, so I couldn't find another figure for pain. So PowerPoint gave me this, but don't use it if you're heartbroken. I mean, we'll put you to sleep, codeine, right? But don't use it. Anyway, so it's analgesics like codeine and capsaicin, that's in chili peppers actually. And then modern mental treatments, treatment metabolites like cannabinoids and psilocybin. And so my group is focusing mainly on actinomycetes. And why is that? So you can find them everywhere in soil. So if you roll yourself to the ground, you'll be covered with actinomycetes. They have this really nice after rain smell that is produced by this compound called geosmin on your left. And because they can be found in soil, we also collect them, and we have an in-house collection of roughly 2,000 strains. And you can see here, my colleagues are collecting strains from Denmark, and I'm here on the right, or your left, in the Black Forest during my external stay in Tübingen. And they're prolific natural product producers and very well-known antibiotic producers. And luckily for us, they produce those compounds from biosteric gene clusters that are genes that are clustered in their genome. So nature does this really helpful thing for us to look at. And yeah, there's just groups of genes that are responsible for the production of a family of compounds. Some of them are unfortunately cryptic or silent under laboratory conditions. Sometimes microbes become domesticated and they just don't want to produce all those things because they don't have to in the lab. So that means that there's a lot of potential and a lot more to be discovered, and that's what we're trying to do. And we use different methods to do that. In the past, our methods were really low throughput in general, from doing this genomics, running our samples, our genomes in anti-smash and intensely looking at the gene clusters for days until we figure out that something is interesting or not. And then the same thing was happening with metabolomics data. So we would do these bioassays. We would test against E. coli or Acinetobacter baumannii, like antibiotic-resistant pathogens. And we would do bioactivity-guided fractionation and separation. We would use like these 15 plates as a first screening to see whether we can isolate something or we see something interesting, or a lot of flasks, because they're also weird growers anyway. I won't get into that. So one example of how low throughput our old method was, was this paper, the discovery and characterization of 2 new macrolides called epimysins A and B. So we looked at the genome, as I said, very intensely for days, and we figured out that there's this gene cluster that looks like a coleximycin gene cluster, which is a known macrolide antibiotic. And we said, we were the chemists of the group, so they told us, hey, take a look at it. If you find something interesting, let us know. So we extracted hundreds and hundreds of plates, and it was a 6-month intensive laboratory labor to finally isolate the molecules. But also the fact that at the very beginning, the whole identification was just extremely difficult, to look at the whole chromatogram and go peak by peak to see whether we can find something that matches a bioactive molecule. So now my group is aiming to build more high throughput workflows for genome mining, engineering of producers, cloning strains, and of course also screening actinomycete strains. And that's where my PhD comes. So I aim to develop a semi-high throughput workflow. I won't say high throughput because it's, it's too flattering. And I think it's, since I'm not using too many robots and there's still manual interpretation, I don't think it's completely high throughput. So thankfully, my team also developed this BGC flow. The publication is coming up. So first of all, we have high quality sequencing, genome sequences of about 1,200 strains right now out of our collection. And my team built this workflow called BGC flow. Again, has big scale and a lot of tools. And in the end, what I'm getting is that this magic table with biosynthetic gene clusters, and if they're known or unknown, what they might produce, etc. So that gave me the opportunity to very easily go into these data frames and pick 100 genomes that I thought that they were really interesting or might have a potential. And I developed this experimental workflow, like more microscale fermentations. I used 24-well plates with beads. Anyway, I won't get into that. And finally, I acquired 1,250 raw data from our Thermo-Orbitrap IDX instrument. And of course, the question was what to do afterwards. That took most of the part of my PhD. And that's why I turned to OpenMS, which, as the name says, it's an open source library of tools and workflows for proteomics and metabolomics. It's vendor independent, so you can use it with MZML or MZXML files, and it's written in C++, which is super fast. But I didn't know programming when I started. So luckily for me, they wrapped everything into Python. in what they call PyOpenMS. So I could just use only Python, or I was promised that I would only look at Python, but that wasn't true. So yeah, and on top of that, they're very, very responsive. They're really amazing people, very helpful. So yeah, I started tailoring this this workflow that became more and more complicated over the months. It's called now UMetaFlow. The paper is almost out. It's a non-targeted metabolomics workflow for high throughput data processing and analysis. So again, it's open source since I'm using OpenMS and open source tools in general. It's pretty easy to set up. It's compatible with all operating systems. It does rapid processing, as the title says, and you have two options. So I started writing it, as I said, in Python in Jupyter notebooks using PyOpenMS. But then Jupyter is not that fast, of course, so I wouldn't call it high throughput. So I put everything, I implemented everything in SnakeMake using the command line interface of OpenMS. And besides OpenMS, I'm also using Sirius and CSI Finger ID. And also, of course, a lot of Python scripts to integrate this data. And also a tool that was built by OpenMS after my nagging, I think, or at least yeah, developed a little bit on the way to export all the files necessary for feature-based molecular networking and ion identity molecular networking. So if we look a little bit closer to the workflow, now there's a lot of things that you will talk about also tomorrow in MZMine, because more or less it's the same steps that we're following, except a few. Yeah, so initially I'm using this executable called Thermo Raw File Parser, which you can, of course, optionally use if you have Thermo files. If you don't, you have to independently convert them through MS Convert, which is a very popular tool. I just didn't want to add that in my workflow because it's dependent on, so you need to have Windows to use it, right? And then I want it to be more universal. So the first step that you will see in most of the untargeted metabolomics workflows, or all of them, is feature detection. I'm sure the MZMine team will talk about it thoroughly tomorrow. But of course, it detects mass traces that are of identical mass to charge and retention time. It deconvolutes peaks that have local minima and maxima. So just the typical deconvolution that we talk about as chemists. And then it assembles single mass traces into a feature. So really data reduction and feature detection and filtering. And here, very important things to add is your parameters. So you have to know your instrument, you have to know your data and see where is the noise threshold and how your data looks like, so you can, so you won't remove important metabolites, basically. Cool, then map alignment, another very important step. You will also use that tomorrow with MZMine. So here, this algorithm corrects for retention time shifts. That is something that is very common if you use liquid chromatography in front of your mass spectrometry. The algorithm will take the file that has the largest number of features, and that would be your QC, your pulled QC, as Martin said. So if you have a pulled QC, you don't really have to define it. Then an optional step is requantification, is something we added with a lot of work in general. So feature detection generates some missing values, of course, in your data, and we try to tackle that. Gap filling is an option also. Imputation algorithms is another option. But what we did is that we used another feature detection algorithm that will parse through all your raw data and only extract features that have missing values along your data. So you'll have a table, a library of features in the end after map alignment, and it will take the features that have missing values and reiterate in your initial raw data before the feature detection. It is computationally expensive, but it's really nice. So then we have ADAX annotation, and exactly like the name says, this algorithm annotates ADAX and is another reduction step, data reduction step, because it will connect different adducts to the same metabolite, right? Then we have feature linking, so that will cluster all the MS1 information to a single file, a single consensus file. And then we have the GMPS export tool that will generate all the data that you need to start a job in GMPS feature-based molecular networking. Now, because we're using adduct annotation, you can also use the new extra step that's called ion identity molecular networking. Maybe some of you have heard of it. No? I see a little, maybe. Okay, so this, anyway, a GMPS export will generate an extra table here if you have adduct annotation. And this basically feature-based molecular networking, what it does is that it will find matches in MS2 level. And sometimes there will be networks that will remain unconnected because the different adducts fragment very differently. So if you have also the MS1 connection, then you will have information reduction, and there you have networks being connected just because of the adduct annotations. Cool. Then we have Sirius and CSI finger ID. So these are formula and structural predictions. And when this job is done, of course, you'll have a table of like very nice table with your master charge, retention time information, the intensities in every single sample, and the Sirius and CSI annotations. And then if your GMPS feature-based molecular networking job finishes, if you add a couple of files in the workflow, that would be your graph ML, the cytoscape file, as well as the annotations, the MS2 annotations, so the matches that GMPS will make with the public libraries. Then you'll also get this GMPS IDs, ID column. So it's like a complementary identification. And then finally, adding your GraphML would mean that in the workflow, you can just integrate the Sirius and CSI finger ID annotations in the cytoscape file for visualization. Cool. That's all. I also evaluated the workflow, of course, using in-house data to begin with, and that's why I built it. So I used about 30 files that I know some of them were commercial standards, some of them are actinomyces that are known producers like model strains, and the results were really, really good. So I got 100% of the features detected, about 76% formulas accurately predicted by Sirius, and they are modest. So they only say that about 70% will be, maximum 70% will be accurately predicted. And then about 65% of the structures accurately predicted by CSI finger ID. Now, just to letting you know, Sirius and CSI finger ID will be presented on Wednesday. Thank you, by Kai. So you will learn about it. Good. And then I thought that it's a little bit biased to only test my own data, of course. So I took this benchmark data set called NTMSS2022, developed by this group. Of the publication is hidden, but yeah, you have access to all the presentations. So what they did is that they took 2 different groups, 5 replicates per group. Both samples, both groups had exactly the same metabolites, but the groups had different concentrations of metabolites. So they used these samples to check the performance of this untargeted metabolomics software for feature detection, quantification, and marker selection. And by marker selection, I mean that 50 of the compounds inside between the 2 groups had massive concentration differences. So your software should be able to kind of figure out the fold changes between the intensities with the intensity information. All right, so they tested Compound Discoverer, which was really, really bad, MS-Dial, XCMS, and the winner, that was MZ-MIND2. And I thought I should also add OpenMS, or the whole workflow anyway, but it was, of course, OpenMS that did the job. And after a lot of parameter optimization, we got out of the 836 true features that were manually detected, we could annotate 797, which was a great score, about 95%. And then from this number, about 95% accurate quantification. And finally, we could detect 48 out of the 50 true discriminating markers. So they did a pretty good job, I think. So yeah, all in all, I hope that I constructed a universal platform for fast replication of LC-MS and MS2 metabolomics data, and hopefully minimize the time and consumables needed. And my team aims to also connect genomics with metabolomics because of all the high quality genomic data that we have. Yeah, thank you so much. Any questions? Any questions? Oh, yeah, please.
Sounds really, really interesting. I really like it. With these validations, however, I always ask, you're the expert in your algorithm. Did you also run the comparison on the other algorithms, or did the experts run it?
No, so
you said you already mentioned after setting all the parameters correctly, you were better.
No, I didn't say we were better. So we were one of the best.
In the end, you came out here as performing.
Yes.
Outperforming the others, and I'm wondering what would happen if an expert in one of these others.
Yes, so
done the same thing. So this is you doing all the algorithms.
Very valid points. Very valid points.
It's not possible to do it differently, but I'm wondering whether that would have made a small change in some of the other algorithms.
Very valid point, and if I had time, I would do it really, but I have to hand in November. So, but you're absolutely right. Thankfully, the authors published all the parameter settings and every single step that they did, all the calculations. Hopefully, they're not lying, but you're right that yeah, it could be a little bit biased. Yeah. All in all, I just want to show that OpenMS is performing really, really well, and it doesn't produce a lot of false positives, and it's just a good tool, and definitely room for improvement as every tool, but yeah, just that it's competitive. Yeah.
It's very nice that it's completely open source.
Yes,
it's very important.
It's very important. It's very important. And there is a graphical user interface if you have a few data called 9. I don't like it personally, but for a small number of data, is also pretty straightforward if you don't know, if you can't code. Yeah.
Can I add something?
Please, who? Oh, yeah.
So yeah, like I'm Robin.
NTMSS2022.
But so I also think like sometimes I guess there's always a feeling, okay, like so many different tools for the same thing, and then there should be some competition, kind of. But I don't feel like it's competition, because also some of the tools, you know, it's good that we have some tools in R and Python and Java and so on.
Yeah.
And it's not like redundant, because people have different skill sets, and there's also different like ecosystems around the different languages and so on. So it's actually good that we have those different options, and you can choose. You can also like plug them together in workflows, which is like the nicest part. Just get out like some standard formats and get it out. And I think those like evaluations, I mean, you just have to do them, right?
Yeah.
Like, you have to show like, hey,
you have to,
protect the stuff that we should protect. But it's just very hard, and you can try to be as fair as possible to like the other software tools, but no one should like over evaluate like and overemphasize the results. You know?
Yeah.
So I think it's great to see
Definitely
that we have this like wide ecosystem.
Maybe it's satisfying to see that Compound Discover did not perform that well compared to the openly available ones, because it's just so expensive and not a lot of people have access. So although in I think the metabolomics conference, they said that they are using now a different algorithm for feature detection. So though this is outdated already for MZ-3, I guess, also, and probably compound discoverer. Yeah. And you're right, it's just always graphical user interface wins, because I mean, if it's also fast, like you're going to show tomorrow, but it's definitely, I understand that it's difficult to learn programming if you're a biologist or a chemist, and it can be really overwhelming, but it's something that everyone should know nowadays, and slowly, just some Python, anything, and have the flexibility of adding extra steps for your pipeline and your data visualization and everything. It's just, it gives you great freedom. But yeah, you're right. Yeah.
Thanks, Eve, for a nice talk. The read ones, can you explain them a little bit?
Yes.
Because there's a huge difference between the final validation.
Yeah, it is. Well, it is not that big actually. If you look at the, no, you're right. It's pretty big. Yeah, just noticed. Yeah, it's pretty big. Okay, so the requantification algorithm basically, after feature detection, you have the feature matrix, right? With all the master charge, retention time information, and then all your samples, like this table here, sample 1, sample 2, with all the intensities. So we will create a library of metabolites that have some non-values. So if there is at least one missing value in your intensities, it will create a new library. We will, well, the workflow will create a new library, and then this algorithm, which is usually used for targeted metabolomics, will take that library and reiterate at the raw data and pick out even low intensity peaks, features that were filtered out by the feature detection algorithm, and yeah, requantify the whole feature throughout all the samples. So it's a little bit different at gap filling, because it will not just fill in the gaps, it will just requantify the whole feature throughout all the raw data. It's just a master charge and retention time match, basically, MS1 level. Yeah. Yes.
Could you give us some details about the fail of compound discovery? What was the workflow there, or what do you think it was the main issue with the fault, for example, false discriminating?
So I think that's a problem of the algorithm, probably of feature detection. So I don't know exactly the settings that, I don't know about the algorithm at all. But what I can see is that the quantification is very off. So they only could quantify for 64% of the features that they managed to annotate, which is also on the lowest end. So this is something that has to do with their the first step of their workflow, which is the feature detection. I really don't know what to tell you. Could be a mass trace error. There could be just that the algorithm starts at the very start of the peak and just ends at the middle of the peak and just quantifies it in the wrong way, or it's really a black box. No one tells you what they're using, or they don't share the information at all, unfortunately.
Another question, if I want to apply this, how dependent is it? I mean, some of these new tools like Sirius say you have to have really high resolution data. How is this with your workflow? How important is it? How relevant is it? Of course, the better the resolution, the better your results will be, but have you looked into that?
So Sirius depends on of course, MS1 data, mass accuracy, but also the fragmentation data. So it would predict formulas and structures dependent on that. But I haven't really checked with, so you have to have high resolution, you're right. And maybe
For your alignment and stuff
I think it is. Yeah, okay, I see. So for the requantification, for sure. Otherwise, you'd get a ton of false positives. Maybe, can you help me, Axel? Maybe feature detection? Could you do it with low res?
Requantification, because that's based on NC and RT match, therefore you need the accuracy.
Yeah.
But before that, you really take the features anyway, right?
Yeah. I think you need probably high accuracy. Yeah. I'll have to look into that and respond again. Sorry. Anyone else? Or should we welcome Martin again? Oh, all right.